Hello, everyone. My name is Lan. I'm a computational chemist by training. I have years of methodology development and drug discovery experience using computational-based method. Currently, I'm working at GHDDI to assist drug discovery project with computational approaches. Welcome to the class. Today, the topic of this class is in silico modeling and the application in the drug discovery against COVID-19. Here's our outline of this class. I will give a brief introduction about COVID-19 and related coronavirus life cycle, which will give potential treatment options. And then I will give the background on the application of computational approaches on the drug discovery in general, and I will specifically talk about protein-ligand interaction and the prediction and its application. Then I will give a demo of the case study using a computational approach to target SARS-CoV-2 3CL protease. As everyone knows, the current situation of COVID-19 is very severe. Based on the statistics reported by World Health Organization, until May 10th, there are around 4 million confirmed cases. And we can see that there's a rapid spread of COVID-19 since January, and especially after February, there's a tremendous increase of confirmed cases. This caused by the pandemic. This disease is caused by a new strain of coronavirus called SARS-CoV-2. You can see the structure of the virus. It has spike protein, which can recognize by the host receptor, enter the cell, replicate, and release. We'll talk about in the next slide. This rapid increase shows that the COVID-19 compared with SARS or MERS epidemic, COVID-19 is more transmissible due to several reasons. One is that the patient can have no symptoms, and the latency period is long, and it's also highly infective. It is also expected that it will stay permanently. There are seasons of peak before the population, before having the population immunity. So there's urgent need of treatments to stop COVID-19 pandemic, although there's potential drugs or treatment in the clinical studies, but we still need a well-designed, randomized clinical trial to justify the efficacy and safety, such as remdesivir, chloroquine, etc. In this slide, I will briefly talk about the coronavirus life cycle and the potential treatment of the options. So here's a figure I took from a Nature Review article published in 2016. This figure just will describe the replication cycle and the related treatment options. The coronavirus can enter the host cell by two different ways. One is through the endosome pathway by binding the receptor binding domain of S-glycoprotein to host the receptor like ACE2, and which triggers conformation change and fusion of the viral and host cell membrane. Another way is by the cell surface non-endosome pathway after the S protein is activated and cleaved by host protease like transmembrane protease serine 2. When the virus enters the cell, it will release the nucleocapsid and the viral RNA used for the translation of the open reading frame 1A or 1B into the large replicase two polyproteins, PP1A and the PP1AB. And these proteins will be cleaved by 3CL-like protease and papain-like protease to produce non-structured proteins. These non-structured proteins then form the replication and transcription complex, allows for the genomic replication of the virus and production of subgenomic RNA, which is then translated into structural and accessory proteins. And then the virion will be assembled in the cell, and the new virus will be released from the host cell and begin another cycle. Here are 3 attractive viral targets, which show actually there are many more viral-based treatment options, which show the red in the figure. One of the viral target I talked about is the 3C-like protease as a demo case. Of course, there are other like host-based targets for the treatment as well, which is shown in the blue color in the figure. So given the current severe situation of COVID-19 pandemic, how the computational approach can assist the drug discovery to identify treatment to really conquer this disease, I will give a brief introduction about how computational approach can assist the drug discovery process in general. So for the stage of target identification and validation, we have methods to identify target. And for the target of interest, we can predict the protein structure using different ways. And also there's methodologies to predict the target druggability. On the stage of hit discovery, one important application is virtual screening to identify new hits. And also we can design a new library using novel chemistry or deep learning based approach. On the stage of hit to lead and lead optimization, we can do rational drug design based on structure or de novo drug design. And we can use there's approach to predict the binding affinity and use for ranking the compounds and make suggestions to Medichem. And sometimes binding kinetics is very important to the cellular potency. There's also approach to predict that. As we know, to making a compound to be a drug, the drug-like properties is very important. And there's a method to predict adenine and tox properties. And also there are mathematical models to predict the PK/PD properties, and this is also very important to making a drug. The next, I will talk about one important factors related to drug potency,